Welcome to the Scientific Dose, everyone. Today, we're embarking on a deep dive into one of the most phenomenal microscopic battlefields known to science, your body's highly specific defense network. We are going to completely decode the intricate and honestly awe-inspiring world of adaptive immunity.
So think about a time you had the chickenpox as a kid, or maybe when you got a vaccine. Have you ever wondered how exactly your body remembers that specific virus a whole decade later? It's a pretty profound question. I mean, you get sick, you recover, and years later, your body somehow retains this perfect, silent blueprint of that exact invader, ready to strike before you even feel a single symptom. That is the magic of long-lived immune memory, and it's exactly what we are going to unravel today.
To make sure we don't get lost on this journey, here is our anatomical map for today. 1. Meet the adaptive immune system. 2. Antigen-presenting gatekeepers. 3. T cells, commanders and assassins. 4. B cells and antibody arsenals. 5. T helper subset and regulation. And finally, 6. The power of immune memory.
Okay, let's just jump right in. Section 1. Meet the adaptive immune system.
Looking at this graphic, it brilliantly captures the entire life cycle and all the hallmarks of an adaptive immune response. If we start right at the top in the purple section, we have antigen specificity. This is where a naive T cell, utilizing these incredibly specific receptors generated through complex DNA rearrangements, first recognizes an invader. And this process strictly relies on self versus non-self discrimination, so it doesn't accidentally attack your own body. Then, moving clockwise into the green section, this recognition triggers clonal expansion. This is a massive, rapid proliferation of these specific cells. Next, moving down into the blue and gray areas, we hit the effector functions. This is where the actual battle takes place, using antibodies and cytotoxicity. And finally, at the very bottom, we see the memory response, where a select few cells self-renew and just hang out in the body, exhibiting tissue adaptation to provide long-term, protective local immunity. It's a perfectly cyclical, highly regulated defense mechanism.
To actually execute these defenses, the adaptive immune system is split into two main branches. On one side, we have humoral immunity. This is driven by our B cells, which basically act as microscopic weapons factories. They secrete antibodies that target extracellular microbes, hunting down free-floating threats like bacteria in your blood. On the flip side, we have cell-mediated immunity. This is driven by T cells, which act more like cellular assassins. They target and kill infected cells that are harboring intracellular microbes, basically eliminating the reservoirs of viruses that are hiding inside cells where antibodies just can't reach them.
But wait, how do these highly specialized lymphocytes know exactly when and where to fight? Well, that brings us to Section 2, antigen-presenting gatekeepers.
Take a look at the skin tissue on the far left. Notice that immature dendritic cell? It's acting like a scout, constantly sampling its environment for microbial peptides using what we call pattern recognition receptors, or PRRs. When it detects a threat, that PRR signaling triggers a massive transformation. The cell literally stops eating, loads those microbial fragments onto major histocompatibility complex, or MHC molecules, and upregulates co-stimulatory molecules on its surface. Now track its physical migration. Following chemical breadcrumbs, it travels out of the skin, straight through the lymphatic vessels, and directly into the lymph node, which you can see on the right, to sound the alarm and activate our naive T cells.
Now that our scout has safely arrived in the lymph node, we reach Section 3, T cells, commanders, and assassins.
What's really fascinating about this lock-and-key process is just how rigorously your body controls it. It actually requires 4 distinct signals. Signal 1 is antigen presentation via MHC, meaning the T cell receptor has to perfectly recognize the foreign peptide on the dendritic cell. But that's not enough. It also absolutely needs signal 2, co-stimulation via CD28 and CD80. Without the second signal, the T cell just becomes inactive, which is a brilliant failsafe to prevent accidental autoimmune attacks. Next up, signal 3 is cytokine secretion, which dictates exactly what kind of T cell it's going to become. And finally, signal 4 is nutrient availability, providing the metabolic fuel needed for the massive cellular cloning what's about to kick off. All 4 of these must be present to ensure it only attacks a genuine threat.
While the T cells are gearing up for close-quarters combat, let's shift over to Section 4, B cells and antibody arsenals. Once activated, B cells class switch to produce different types of immunoglobulin isotypes, and each one has a totally unique effector function. Let's just succinctly break down these antibody classes. IgG is your heavy hitter. It's responsible for the neutralization of microbes and toxins in the blood.
IgA specializes in secretion into tracts, so it's protecting the mucosal surfaces of your gastrointestinal and respiratory systems. IgM is fantastic at activating the complement system, which is this cascade of proteins that literally punches holes in bacteria. But I want you to pay special attention to IgE at the bottom of the table. IgE has a highly specialized job: Helminth defense, protecting you against parasitic worms.
To see exactly how IgE works in action, notice the tiny green Y-shaped IgE antibodies acting as a literal physical bridge here. On one side, they are clamped tightly onto a large red immune cell called an eosinophil. On the other side, they are binding directly to the large purple Helminth parasite. This bridging is the exact trigger that tells the eosinophil to degranulate, which basically means it dumps its toxic, lethal granule contents directly onto the worm, leading straight to parasite death.
So the really crucial takeaway here is that antibodies don't just act in one single way. They have multiple effector mechanisms. They can perform neutralization of microbes and toxins, essentially physically blocking a virus from ever binding to your cells. They perform opsonization, which is basically coating a pathogen to make it super attractive, almost like making it tasty, for massive eating cells called macrophages to devour whole. And importantly, they trigger classical complement activation to lyse, or burst open bacteria.
Now, to coordinate all these different weapons, we definitely need Section 5, T helper subsets and regulation.
Based on the specific chemical signals they receive from the dendritic cell, a naive CD4 plus T helper cell will differentiate down a very specific path to handle a very specific threat. Look at the top path, Th1. Th1 cells release the signature cytokine interferon gamma, which powerfully activates macrophages for host defense against intracellular microbes. However, misfires here are a primary driver of autoimmune diseases. The middle path is Th2. These release IL-4 and IL-5 to fight helminthic parasites, but they are also the primary culprits behind allergic diseases. The bottom path is Th17, which uses IL-17A to recruit neutrophils to destroy extracellular bacteria and fungi, while also playing a major role in various autoimmune conditions. It's an incredibly customized response system, but obviously, it comes with very high stakes.
With all this lethal, custom-tailored firepower, we absolutely need a way to hit the brakes. Enter the green regulatory T cells, or TREGs, marked by the FOXB3+ protein shown on the far right of this lineage tree. Driven by cytokines like TGF-beta, these cells don't attack pathogens at all. Instead, their entire job is to secrete suppressive cytokines. They are absolutely crucial for maintaining peace, enforcing peripheral tolerance, and ensuring that once the infection is totally cleared, our lethal defense is powered down before they cause devastating autoimmune damage to our own tissues.
Let's examine the biological aftermath in our final chapter, Section 6, The Power of Immune Memory.
Tracing this timeline from left to right, we start with the naive state, just a few specific cells waiting around for their perfect match. Upon activation, we see this massive, steep upward curve, the explosion of clonal expansion during the effector phase, where millions of cells are drafted for war. But look at what happens next. The infection is cleared, and we see a very sharp drop-off. This is clonal contraction, where the vast majority of these effector cells undergo apoptosis, programmed cell death, simply because they're no longer needed. However, the line doesn't go all the way back to zero. Notice that vital elevated plateau on the far right? These are your long-lived memory cells. They remain in your tissues and bone marrow for decades, providing immediate, overwhelming force if that specific pathogen ever dares to enter your body again.
Which brings us to one final, really thrilling thought. We've seen the sheer elegance of antigen presentation, the precise lock and key activation, the tailored antibody arsenals, and the perfect balance of regulatory cells. If our immune system is naturally this sophisticated at recording and remembering every single threat it encounters, what incredible diseases, from rapidly evolving viruses to treatment-resistant cancers, could we cure by fully mastering the programming of our own immune memory? The future of medicine lies exactly in decoding this microscopic defense network. Thanks for joining me on this deep dive. Keep learning and stay curious.